Hey everyone, we are going to be concluding our lipid lectures today with the following topic, so buckle in for cholesterol metabolism. Our learning goals will be to illustrate cholesterol's biosynthetic pathway and then to define the pathways of cholesterol trafficking once it's made throughout the body. I'm going to blow your minds when I say that our molecule of the moment is going to be cholesterol. Take a look at its molecular structure. It's a complicated beast. I'll remind you we've got 4 different rings of carbon fused together, plenty of chiral carbons, tertiary carbons, secondary carbons with a very specific geometry. This is a hard molecule to create. So it stymied scientists for a long time on how biology managed to do it, frankly. The biggest clue that uncovered the biosynthetic root of cholesterol came from this very famous experiment called the acetate experiment. Now, how did this acetate experiment work? Well, the first thing scientists did was make a very expensive batch of acetates, all right? Normally, that's a cheap molecule. However, this isn't just your garden variety acetate. Take a look at this carbon. I've colored it red. It may be hard to see, but that's indeed a red colored carbon. And we're coloring that CO minus carbon red for a good reason. It's isotopically labeled. We used a special heavy carbon isotope, 13C, and inserted it into that position. And scientists can do experiments to detect the presence of this label. And that's not the only label we put in. We put a different label on the hydrogens associated with the methyl attached to that acetate. And we didn't give it a 13C label, we gave it a different deuterium label, a heavy hydrogen H2 label. And we're going to color that black. All right? So we're going to take this expensive bit of acetate and feed it to some rats. And then over time, the rats are going to incorporate that into their bodies enzymatically. And we extracted cholesterol from those rats and discovered a very impressive isotope distribution. Take a look again at this cholesterol molecule, and I want you to pay attention to the arrangement of the red carbons along with the black carbons. I want you to study this molecule and then step into the shoes of the scientists that did this acetate experiment. Predict what this experiment implies about the biosynthetic pathway of cholesterol. There are many correct answers to this question, and I'm looking forward to hearing one of them in class. The first goal for today's lecture will be to survey how biology takes a simple unit like acetate and makes it one of the most synthetically challenging natural products out there, namely cholesterol. And as challenging as it is, all animals are capable of making this compound. Doing so is going to be very laborious. It's going to cost several ATP molecules, free energy, along with several molecules of NADPH, aka reducing power, the same kind used to make fatty acids. And we can basically divide cholesterol biosynthesis into 4 general phases. The first of these 4 phases is called condensation. The goal of this step is to take 3 acetates and condense them into a special molecule called mevalonate. You can see that over here, mevalonate. The second step is called activation. Here we are going to activate mevalonate By converting it into something called activated isoprenes. Step 3 of cholesterol biosynthesis is going to be called addition. Here we will take 6 isoprenes and connect them together into a special precursor compound called squalene. And lastly, this is like the crazy step, cyclization. We are going to take our squalene molecule and scrunch it up and then do a series of cyclization reactions to form what's called the steroid nucleus. That's like the backbone of cholesterol and all sorts of cholesterol derivatives. This here, by the way, is what we specifically refer to as the steroid nucleus. Condensation, activation, addition, cyclization. There you go. Let's dive right in with cholesterol biosynthesis. The condensation steps will begin with the enzyme acetyl-CoA acetyltransferase. The job of this enzyme is to catalyze the condensation of two acetyl-CoA molecules, specifically in a head-to-tail fashion. One side of one acetyl-CoA molecule will link up with the opposite side of another. And you can see how in the product of this reaction, acetyl-CoA, it looks like we just stapled together two acetyl-CoA units. This is one, and this is the other. We have to, of course, remove coenzyme A as a leaving group to facilitate this condensation. The second reaction of the condensation steps is catalyzed by a different enzyme, HMG-CoA synthase. This is also going to facilitate a condensation, specifically a Claisen condensation, but here we are doing a tail-to-tail connection of our freshly made acetyl-CoA and another acetyl-CoA. And what we're going to do is take the tail of our acetyl-CoA and the tail of this acetyl-CoA and connect them. That's going to turn this into an alcohol, which you can see over here. And then this red portion comes from the acetyl-CoA that we just tacked on. And the product of this reaction is a bit of a mouthful, beta-hydroxy beta-methylglutaryl-CoA. We just call this HMG-CoA for short. If these reactions look familiar to you, by the way, that's a good thing because this entire process happens to be chemically identical to the first half of ketogenesis, aka the starvation pathway. This next step in condensation is extremely important because it is here we will diverge from the catabolic ketogenesis route and head into the anabolic cholesterol route with the enzyme HMG-CoA reductase. This is the most important enzyme in the cholesterol synthetic pathway. This is the committed step. It is a major regulatory points in cholesterol biosynthesis, and therefore it is also a very druggable points in the pathway. How does this enzyme work? We are going to take HMG-CoA and reduce it using 2 equivalents of NADPH. We are going to use this reducing power, and we are going to get rid of the coenzyme A and turn this thioester into a primary alcohol. This alcohol that we just made, mevalonate, is highly important because it is going to be committed to anabolism. It is the mother of not only any steroid, but also any isoprenoid. So cholesterol, yes, will be derived from mevalonate, but also other important compounds like chlorophyll, carotenoids, vitamin A, ubiquinol, coenzyme Q, all of these extremely useful lipid derivatives that are not fatty acid based. Once we make mevalonate, we are ready for step 2, activation. Our goal here is to destabilize mevalonate so we can anabolize it into a larger precursor compound. Mevalonate 5-phosphotransferase. This enzyme is going to phosphorylate the primary alcohol we just made using an ATP molecule. Remember that phosphates are going to be good leaving groups. So this alcohol will get phosphorylated by this ATP to give 5-phosphomevalonate. The 5th enzyme in this pathway, phosphomevalonate kinase, is going to do the same thing again, basically. We're going to upgrade our phosphate group with another equivalent of ATP and turn it into 5-pyrophosphomevalonate. If 5-phosphomevalonate was a good leaving group, pyrophosphomevalonate is an amazing one. The next enzyme is a little bit messy. The goal of pyrophosphorylmevalonate decarboxylase is twofold. The first step is going to basically involve another phosphorylation. We will take ATP and attach it now to the other alcohol in this mevalonate backbone over here, shown in red. This phosphate is going to serve as a good leaving group. And we need that leaving group to facilitate a combined beta elimination and decarboxylation reaction that I'm going to show next. Because our goal here is to turn all of this into a terminal alkene. On the next slide, I'm going to show you how we can remove this carboxyl group to facilitate the formation of an alkene. I will now show you how this combined beta elimination decarboxylation occurs. And remember, this is still the same enzyme we left off with last slide. It's all pyrophosphorylmevalonate decarboxylase. That enzyme that just installed this phosphate group on the alcohol over there is going to next pry off this COO minus. It's going to cut the bond over there and release it as CO2 gas, shown over there. Since we are generating gas, this is going to be a very entropically favored reaction because generating gas is going to generate a lot of disorder. Now, the electrons in the bonds that we just cut, highlighted over here, they need to go somewhere. So we are going to relocate them to this carbon-carbon bond and turn it into an alkene. However, we can't have bond order of 5 on the carbon next to it, though, which means one of those other 3 bonded groups needs to break if we're going to make this allowed. And looking at it, the only bonds that we could cut that would release a sensible leaving group is this one. Cutting that bond is going to release phosphate, shown over there. So when all is said and done, 3-phospho-5-pyrophosphomevalonate becomes delta 3 isopentyl pyrophosphates. We call it delta 3 because on the 1 to 3rd carbon, there's going to be a double bond on this isopentene unit. And this IPP molecule is the first activated isoprene we see, and it's activated thanks to this pyrophosphate moiety, which is, again, an amazing leaving group. The next enzyme to work on this activated isoprene is isopentenyl pyrophosphate isomerase. What this enzyme will do is a hydride shift, and it's going to rearrange the location of that terminal alkene and move it inwards. And we do that by grabbing one of these hydrides and moving it to the end. And in doing so, that's going to reposition the double bond to go over there, turning IPP into the next intermediate called dimethylallyl pyrophosphate. We sometimes call this DMAP for short. We're almost done making cholesterol. Moving on to step 3, addition, our goal now is to link up these activated isoprenes into a really long terpene. First, we need the enzyme prenyltransferase. This is going to facilitate a head-to-tail condensation of 2 different activated isoprenes, DMAP and IPP. Remember that IPP has the terminal alkene, and DMAP is basically IPP that underwent that hydride shift. When we connect together DMAP and IPP, we are going to get a 10-carbon compound called geranyl pyrophosphates, or PP for short, shown over there. And you can see that we just stuck together one of these and one of those into that. And we lost one of our amazing pyrophosphate leaving groups in the process. But we still have one more, which we are going to utilize again with the same enzyme, prenyltransferase. It's going to keep on working, and it's going to take the geranyl pyrophosphate and stick on another IPP, and it will also do so in a head-to-tail fashion. The product of this condensation, of adding in this unit over there, is going to be farnesyl pyrophosphate. Adding in this 5-carbon unit to the 10-carbon geranyl pyrophosphate will give us a 15-carbon intermediate. Squalene transferase is the third and final enzyme involved in the addition series of reactions. This is going to catalyze a head-to-head condensation between two farnesyl pyrophosphates. And this is what I mean by that. Let's establish this side of the molecule as the head region. We're going to take them together, chop their heads off, and stitch their bloody neck stumps together to form this squalene. And where I've highlighted is basically where those two pyrophosphates used to be. And we need a little bit of NADPH to facilitate this process. So it indeed requires reducing power. But in total, we're connecting two 15 carbon compounds together, giving us a 30 carbon molecule called squalene. In the last phase for cholesterol biosynthesis, our goal is to reactivate squalene so it can undergo a series of complex cyclization reactions, taking us from a symmetric molecule to a highly chiral or asymmetric one. The enzyme squalene monooxygenase is going to perform a regiospecific epoxidation upon one of the distal alkenes. Let's say this one. This process is going to be a redox process. It's going to involve oxidation and a reduction. We will need an equivalence of NADPH. And in doing so, we're going to generate a very powerful electrophile. And we need this to enable squalene to do something pretty cool in the next step. The next enzyme is going to take our squalene 2,3 epoxide and wind it up into this interesting zigzaggy meandering configuration. And then oxidosqualene cyclase is going to perform one of the most complicated mechanisms in all of biochemistry. If you ever need a migraine, I highly recommend you Google it because, oh boy, it's a doozy. I will not be going into that level of detail in this course. All I want you to know is that it's going to use a lot of catalysis by approximation to wind up squalene 2,3 epoxide and then facilitate these alkenes that we installed into it by our activated isoprenes into a series of 4 ring closing reactions, like so. We're going to have this pi bond at the very end, or almost the very end, come in and form a sigma bond there. That's going to cause the next pi bonds to rearrange like so, and then we get this chain reaction all the way down the length of the molecule. And these four ring closures are of course enabled by this powerful electron withdrawing effect installed through this epoxide that we installed in the previous step. So while we do these four ring closing reactions, we're also going to be generating a lot of stereocenters in the process. And that's what makes cholesterol such a great backbone for all these different kinds of hormones. Multiple stereocenters means that we can put many substituents there in a highly modular fashion. So simple changes can result in great differences in molecular geometry, which means it can fit into different receptors. And that's a good quality to have when you're making a library of hormones. Now, here's the final situation. When we have oxidosqualene cyclase do this really elegant reaction, the product isn't cholesterol. It's actually something called lanosterol. So that is the direct product here. And I know this is a little like your princess is in another castle, but lanosterol through a multi-step process, I'm talking like 20 more reactions, will eventually become this coveted cholesterol molecule that we've been talking about. At the end of the day, what I want you to appreciate is that cholesterol biosynthesis is one, complicated, and two, it's going to be very expensive. Let's do some brief accounting so that we can catch up with what we invested into this cholesterol molecule. Each isoprene activation is going to cost us 2 NADPH units and 3 ATP molecules. And remember, we're going to need 6 activated isoprenes in order to make this squalene molecule that we're going to wind up into lanosterol, which will eventually be converted into cholesterol. So minimally, we need 12 NADPHs to make this critical substrate. Don't forget that squalene monooxygenase is also going to require an NADPH. And then we'll need 18 ATP total because 3 times 6 is going to be 18. Once you have cholesterol, there's many ways that biology can further fine-tune the molecule to yield an array of functionalities. For example, if you were to oxidize cholesterol, you would get bile acids or bile salts. This is actually how you excrete spent cholesterol. We can't break down cholesterol like you would a fatty acid or a glucose molecule. So instead, we're going to tack on a highly charged group, such as the sulfone, and basically convert the cholesterol molecule into a soap molecule. We're going to pump that into our intestines with every fatty meal, and that is going to help emulsify dietary fats. Now, some of the bile salts will, of course, be taken up by our intestine, but a lot of it is also just going to pass through with our stool. So that's how we get rid of cholesterol, basically. Now, here's a fun fact. Dietary fiber, which is basically cellulose, is often celebrated as being heart healthy because it can glue up and sponge up the bile acids our gallbladder secretes, and that's going to reduce the efficiency of fat absorption and therefore bile reuptake. So dietary fiber is going to impede dietary fat absorption. And that's why it's considered to be a heart healthy component to someone's diet. Now, bile acids are just one derivative of cholesterol. We also have the steroid hormones. These, I think I mentioned a while back, are all cholesterol derivatives. And more specifically, all steroid hormones come from this particular compound called pregnenolone. And we get this from cholesterol by cleaving off the alkyl tail at the D ring of the steroid nucleus. And the steroid hormones, they're going to regulate physiology. They are going to do so many different things. You've got your sex hormones, you've got your stress hormones, you also have plenty of hormones that are involved with water retention. These are all going to be steroid derivatives. The last metabolic fate of cholesterol I'll be discussing here are the cholesterol esters. These may be more of a novel concept to you. Cholesterol esters are actually tagged cholesterols. And we tag them by forming an ester with a fatty acid. When we tag a cholesterol molecule with a fatty acid, what we're actually doing is earmarking this cholesterol for fat trafficking. So, fat trafficking. And when I say fat trafficking, I mean relocation or long-term storage. And the reason why this fatty acid is able to do that, it's because the fatty acid itself is geometrically incompatible with the lipid bilayer. It is too large and way too hydrophobic even to exist within the lipid bilayer membrane, which means it's going to have to gather elsewhere. And that elsewhere is going to be the beginning of fat trafficking. This is where lipoproteins come into play. Recall that lipoproteins are a class of amphiphilic proteins that solubilize both triglycerides and cholesterol esters by wrangling them into these nanoparticle structures. One example of a lipoprotein particle is the chylomicron. And another one that you may have heard of is LDL, low density lipoprotein. And what do these particles have in common, for example? Well, the exterior is going to contain these apolipoproteins. And what these proteins are going to do is recruit and stabilize a phospholipid monolayer to gather around a fat payload. These proteins are going to facilitate the formation of this. So we have a polar exterior, which stabilizes the particle in aqueous blood, and a nonpolar interior, which we can load up with a fatty payload. Think of these lipoprotein particles as burlap sacks filled with triglycerides and cholesterol esters, and we can distribute these burlap sacks to the appropriate tissues. You may also be broadly aware that lipoprotein particles come in varying sizes and densities. The largest and least dense of these are actually the chylomicrons. Largest and least dense. We can see that by examining these electron micrographs shown over here. We only need 60K magnification to visualize a chylomicron. We have to triple the magnification to make the VLDLs, LDLs, and HDLs all visible. And even so, the HDLs are barely discernible from the background. So the chylomicrons are the largest and least dense. HDLs are the smallest and most dense. And if you've ever worked at a blood lab, you can often predict when the lipid panel will be abnormal. And you can tell because the lipoproteins, when they're in abundance, will separate out over time and form this nice, fatty, creamy supernatant on top of the blood tube. This here is a normal patient. And this here is a hyperlipidemic patient. Here the red blood cells have separated from clear plasma. The plasma, however, in this patient is overly enriched with these lipoprotein particles, and therefore it is opaque and very fatty. As alluded in the previous slide, the different classes of lipoprotein particles are different because, in part, they have different densities. They can be separated out into discrete layers through ultracentrifugation. And there is actually a reason why the different lipoprotein particles have a spectrum of densities, and that's because they have different compositions. Let's take a look at this table down here. And we'll start by examining the lower density particles. These tend to be very rich in fat molecules. Chylomicrons and VLDLs, for example, are almost or mostly triglycerides. LDLs, aka bad cholesterol, has a large fraction of cholesterol esters. The high density lipoproteins are actually mostly protein, and hence they have greater density because fat is less dense than protein. These differences are important because these lipoprotein particles have different functions, different destinations across the body. And the instructions on the packaging of these particles, the postage, so to speak, is actually the apolipoproteins that help form them. In this table over here, we have a list of different types of apolipoproteins and which kinds of lipoprotein particles they are associated with and their functions. Some functions, we really don't know what they do. Some other functions help give the delivery instructions to these lipoprotein particles. For example, ApoB100 is going to be a apolipoprotein whose job is to bind that particle to specific LDL receptors or tissues that are programmed to receive cholesterol esters, for example. I don't expect you to know all of the apolipoproteins and what they do. The important ones we'll mention in the following slides. Just be generally aware that there are many and that they serve as postage for the lipoprotein particles and all the contents they contain. My goal now is to talk about fat trafficking, which is the way that the body utilizes these lipoprotein particles to direct cholesterol traffic throughout the body. And there's four different pathways that I want you to be aware of, and we're going to start with the exogenous pathway. This pathway is the way we direct dietary cholesterol that we consume and absorb it into our liver. And this in itself is a four-step process, roughly, and I'll describe it below. Step one is going to be the synthesis of chylomicrons. So synthesize chylomicrons from ingested fats within the enterocytes. And those chylomicrons then enters the bloodstream. And here is a visualization of this process where we have a stream of ingested fats entering the intestine. Then a combination of mechanical and chemical digestion is going to break apart those fat globules into basically a mixture of fatty acids and cholesterol. These compounds will migrate into the cells lining the intestinal wall, and it will all be packaged up into chylomicrons, which are then transferred eventually into the bloodstream. So technically, there's no new information in this first step. This is basically the fat digestion process we discussed during fatty acid catabolism. Step two is where new things are going to happen. The apolipoprotein ApoC2 is secreted from triglyceride sinks, namely, but not always, adipocytes. This ApoC2 will activate lipoprotein lipase, LPL. That is going to begin dismantling our chylomicron, and it will drive free fatty acids, FFAs, into the sink tissues. What we are doing here in step two is selectively degrading the chylomicron. We are going to have our adipocytes leach out preferentially the triglycerides, causing the leftover chylomicron to concentrate the cholesterol content. So step three, the cholesterol-rich chylomicron remnants, we'll call it, migrate to the liver. So this spent portion is going to go all the way to the liver, where lastly, receptors on hepatocytes will bind ApoE, which is located on the exterior of the chylomicron, and it will prompt cholesterol uptake. I'm going to boil this entire process down even further. This is how the body absorbs fats. 1. Eat the fats. 2. Package and deliver the fats to blood. Step 3. Drop off the triglycerides, which I'll call TGE. Step 4. Drop off the cholesterol last in the liver. There you go. The second pathway is called the endogenous pathway. And here we are going to take both dietary cholesterol that we consume and also cholesterol that we make ourselves, de novo cholesterol, and we are going to package it and send it out to the rest of the body, also known as the milieu. So let's break it down. Step 1 of the endogenous pathway begins in the liver, where we just left off. That cholesterol that we just imported, we are going to package and convert into cholesterol esters using an enzyme called ACAT. So C becomes CE, cholesterol esters. What the liver is going to do next is take the cholesterol esters, we're going to combine it with some triglycerides, TG, and then package into VLDLs. Of note, we have ApoB100 on the exterior of the VLDLs, along with some other lipoproteins. But ApoB100 is important. You'll see why soon. So that's where step 1 ends. Step 2 is simple. We're going to kick them out. We're going to export the VLDLs into the bloodstream for extrahepatic delivery. Step 3, the VLDLs are going to circulate and they're going to pass by sink tissues. And these sink tissues are going to release gradually lipoprotein lipases. Sink tissues release LPLs, and that is going to offload the triglycerides first. Release LPLs. And as we do that, we are going to remodel or change the VLDL. First into IDLs, intermediate density lipoproteins, and then LDLs, low density lipoproteins. So as we remove the triglycerides from these lipoprotein particles, the cholesterol ester content is going to concentrate. Step 4, now that we have these LDLs that are super rich in cholesterol esters, extrahepatic sinks will bind ApoB100, which is on the exterior of what used to be VLDLs, but is now just LDLs. It's going to bind those special apolipoproteins, and it's going to induce receptor-mediated endocytosis. We're going to absorb and pick up those bags of cholesterol esters right into the tissues that are asking for it. Any LDLs that aren't grabbed are going to be redelivered back to the liver. So step 5, remaining LDLs return to liver and are endocytosed there. The reverse cholesterol transport pathway refers to how the body ferries extrahepatic cholesterol back to the liver. That's why we call it reverse transport, because it undoes endogenous transport. The first step of the extrahepatic process begins with the secretion of protein-rich HDLs. Liver secretes protein-rich HDL. Step 2, what we will call nascent or freshly secreted HDL matures by scavenging cholesterol esters from either VLDLs or chylomicrons. In the third step, maturing HDL will scavenge cholesterol esters from extrahepatic sinks. This can be macrophages, this can be adipocytes, a bunch of things. Once we have filled up our HDLs with cholesterol esters, we consider them to be mature. HDLs. And these are going to return to the liver, and they will be taken up via a protein called SRBI. So this is how we go grab the cholesterol that's over here, and we bring it all the way back over there. This is why HDL is called good cholesterol, by the way, because it's going to hunt and gather cholesterol from the body. LDLs are bad cholesterols because they're going to distribute cholesterol throughout the body. Lastly, the enterohepatic pathway is how the body will dispose of cholesterol. It's going to begin in the liver, where surplus cholesterol is oxidized by hepatic peroxisomes, and it is made into bile. Step 2, the bile that we just created is going to be transported and stored into the gallbladder. Step 3, bile is secreted into the small intestine, after you consume fats. Step 4, while we emulsify those fat globules and take them up through the small intestine, we are going to have partial reuptake of bile, which again is oxidized cholesterol, and then we can certainly reuse that bile and move it back into the gallbladder. But a good amount of that bile is going to just pass through and be eliminated as stool. No beta oxidation is occurring here. Cholesterol is never a fuel source. Let's briefly rewind to the endogenous pathway. Remember that the mechanism by which LDLs deposit their cholesterol esters into target tissues, sink tissues, is through receptor-mediated endocytosis. In normal homeostatic conditions, this endocytosis is made possible when our said target tissues make and present special LDL receptors. So our rough ER makes LDL receptors, shown over there, and through the endomembrane system, it's going to be delivered to the cell surface for presentation. Okay. Once we have our LDL receptors on the outside, they are going to be on the lookout for any LDL particles swimming by. And we are going to catch, grab, and reel them in by the ApoB100 lipoprotein. So the LDL receptor binds ApoB100, and that's going to result in internalization. Once we have internalized the entire LDL particle, it's going to migrate through the cytoplasm until it collides and merges with a lysosome. And the lysosome is the digestive organelle of the cell. It's going to contain several lytic enzymes that will degrade the entire LDL particle. It's going to break apart the ApoB100. It's going to digest the cholesterol esters into fatty acid and cholesterol. And we are going to ultimately release the contents of this particle into the cell for general use. The amino acids, any triacylglycerols that hitched along for a ride, and of course, free cholesterol. Now, there are two possible ways for this endocytosis process to break down. In Niemann-Pick disease type C, the proteins that are responsible for lysosome trafficking stop working. This causes the cells to take in fats normally, but these fats are going to remain trapped inside of these endosomes. This means the cholesterol esters can't get liberated and used as regular cholesterol. This is going to be bad news for many cells, particularly those with interesting dynamic membrane structure, such as neurons. The symptoms of this disease, therefore, it's going to primarily manifest as progressive neurodegeneration from an early age. So we sometimes call this disorder childhood Alzheimer's. Because it disproportionately hurts the neurons at an early age. Another weak point in this process resides within the anatomy of the LDL receptor itself. Mutations to this receptor are actually somewhat common in human populations. The more mutated this LDL receptor is, the less efficient the entire endocytosis process becomes. This will predispose the individual to have higher levels of LDL in circulation, and that, of course, can lead to atherosclerosis and all other sorts of pathologies. We call this familial hypercholesterolemia, aka mutant LDL receptors. These people, they can have extremely healthy diets, but their cholesterol can still be through the roof. Now, what are some good strategies we can use to protect ourselves from having too much cholesterol in circulation? Well, the best defense to that is going to be a combination of an active lifestyle and a good diet. What this is going to achieve is a high HDL to LDL ratio. A good diet and activity will increase your good cholesterol, the HDL, which is responsible for reverse transport, the mopping up and scavenging of cholesterol from our arteries and tissues, and then we deliver them to the liver for destruction. And that's also going to reduce the amount of LDL, the amount of endogenous pathway that we do, which distributes cholesterol to our tissues and then eventually to our arteries. So these are tried and true tactics to maintain a healthy cardiovascular system. How do we maybe protect people, though, that have familial hypercholesterolemia? They can do everything correctly, but they could still have an unideal lipid profile. Or what about people that are physically incapable of exercising? How do we help them? A very important and useful strategy is statin therapy. Statins are a widely prescribed class of drug that serves as an HMG-CoA reductase inhibitor. If you recall, this enzyme controls the committed step for cholesterol synthesis. If you look at the portion of a generic statin shown here in red, it bears a structural similarity to its product mevalonate. So statins, what they're going to do is gum up a key step in cholesterol synthesis. Statins overall are highly safe drugs, and some of their side effects are actually beneficial to people, such as lowering blood pressure. That's a common side effect. And that's actually a good thing because most likely, if you're being prescribed statins, you probably already have high blood pressure to begin with. Statins do also have some not so nice side effects, of course. All medicine does. Muscle pain and weakness is certainly going to be problematic if you're trying to shift to a more active lifestyle while you are beginning your statin therapy. And extremely rare is rhabdomyolysis or muscle death. And the side effect that most people are apprehensive about is insulin resistance. Insulin resistance can ultimately lead to diabetes. And this has only been observed in certain statins, and even then, there's some debate if it's the statins or the lifestyle that you had before it is what ultimately made you insulin resistant. But still, it's a concerning correlation. I'm not going to downplay it. However, evergreen medical advice that I've been told is never let a potential side effect scare you away from a drug prematurely. Talk to your doctor about it. They're going to be the one that can best advise you and only you. Statins overall, though, have been very good for most people that need them. They save lives. But at the same token, like I said before, an ounce of prevention is worth a pound of cure. And it would be nice to bypass the risk of any side effects by having a good lifestyle first. Let's wrap up everything by mapping out cholesterol metabolism's regulation. I keep on saying that HMG-CoA reductase is the star of the show. Hopefully here you'll see why. In a persistently fed state, we are going to keep cholesterol synthesis active, and we do this via insulin. When we eat, we secrete insulin. And yes, this will signal for increased glucose uptake. However, it is also going to activate a phosphatase called HMG-CR phosphatase. And what this enzyme will do is it's going to boost HMG-CoA reductase's activity. It's going to keep it active in its dephosphorylated form. And this makes sense because cholesterol synthesis is expensive, and it's best to do it when we have plenty of energy coming in. All right, so how does our body tamp down cholesterol synthesis? As you probably would have guessed, the opposite hormone, glucagon, the hungry hormone, is going to be involved. It in turn is going to actually shut down this phosphatase, which is going to help keep HMG-CoA reductase active. and at the same time having low energy charge, low ATP levels, aka an active state, that is going to prompt the AMPK signaling axis, which is going to directly phosphorylate HMG-CoA reductase and therefore shut it down. Said another way, AMPK puts the deactivating phosphoryl groups on HMG-CoA reductase, and glucagon keeps them on. So let's just sum that up by saying phosphorylation slash low energy charge deactivates HMG-CoA reductase. Cholesterol itself can play a role here. When the concentration of cholesterol plummets in the cell, that is going to increase the concentration of some transcription factors, which will upregulate the expression and concentration of HMG-CoA reductase itself. Which will increase the biosynthetic flux of cholesterol synthesis. When cholesterol is high in the cell, the opposite happens. We will not only stop making this key enzyme, HMG-CoA reductase, but we will actually make a special enzyme that will degrade any remaining active HMG-CoA reductases. So not only is the activity going down and the amount going down, but the half-life of the enzyme itself, the amount of time it's active, is decreasing. And this is going to choke the amount of cholesterol that we make. Let's examine this regulatory network again, now in the context of statins. By shutting down the enzyme HMG-CoA reductase, we are going to handicap cholesterol biosynthesis by reducing mevalonate production. There are going to be additional downstream benefits here. Lower intracellular cholesterol, thanks to the lower amount of mevalonate, that is going to trigger a little cholesterol crisis within the cell. It is going to be very thirsty for LDLs, which are going to be a source of cholesterol esters. So the LDL receptor presentation rate is going to increase. So when the cholesterol level goes down, the number of LDL receptors goes up. This is going to lower the concentration of LDLs in the blood, because now more of those LDLs are entering the cells that you are starving of cholesterol instead of them dumping it off in the middle of your arteries. And furthermore, by decreasing intracellular cholesterol synthesis, that is going to also reduce the amount of cholesterol esters that you make, which means cells that are affected by statin therapy are going to have a hampered ability on making these LDLs. So we're not only taking in more LDLs, we're putting out less via this endogenous pathway system. So lower LDLs, less endogenous trafficking. Now, this all sounds terrific, and it indeed is, but a word of caution when it comes to statin therapy. Mevalonate isn't solely dedicated towards the production of cholesterol. We also need mevalonate to make terpenoids. Statins are very useful, but they're not a get out of jail free card. There's going to be a need to monitor individuals who are taking statins because our body certainly needs these. And some people can tolerate this minor hit to the terpenoid production better than others. So, like all medicine, we have to consider the good along with the potential risks of the side effects. So let's leave off here with two bonus visuals that you can study from. The image on the left probably looks familiar. These are the combined exogenous, endogenous, reverse, and enterohepatic pathways all mapped out for you in extreme detail. The figure that's on the right tells a similar story in a more simplified, pared down manner. So I think these will both be helpful when you're studying. And that is all I have to share on cholesterol metabolism. Thank you so much for tuning in. It has been a pleasure.